Okay, thanks all for coming today. It's my great pleasure to introduce Professor Gilbert OMENN from University of Michigan. And in fact, I am from University of Michigan too. So he has an amazing, distinguished career for him. And I will give you of what he has done. And please check, please see, there's a, in fact, Dr. OMENN has a Wikipedia site, so that will explain who he is. So his main area for last 7 years about human proteome project. He's one of the, I think, co-founders for human proteome project. And many you can read about him. I highly encourage you to read about him. And so I think he's so many things he accomplished. But today he will talk about computational and bioinformatics innovations in proteogenomics, predicting function of unannotated proteins and facilitating identification of missing proteins. So you welcome, Dr. OMENN.
Well, it's a great pleasure to be here. I was here 2 years ago with Professor Schmale and my colleague, Professor Indika Rajapakse. He just went to visit his parents in Sri Lanka with their granddaughter and his wife, who will be a scientist speaker here tomorrow. And we're very proud to have him in our faculty at University of Michigan. The subject of this month-long extended workshop is genomics, cells, and mathematics. And Professor Schmale and Rajapakse have made a remarkable coalition of effort around bringing mathematical thinking and tools to biological subjects like the gene, like control theory, like cell cycle, like reprogramming of cells. I don't need it. It's a very exciting and refreshing idea to bring first principles to complex biological scientific phenomena. The subject here is not just mathematics, but genomics and cells to be understood better with mathematics. And the message I'm bringing today is to help you make a connection between the information in the genes, in the DNA, and the functions of cells. Cells are what make up our bodies. And DNA doesn't act directly on most functions. If you want to understand DNA functional effects, what's called functional genomics, there are other partners, of which the most diverse and most interesting, I would say, are the proteins. So there is a flow of information from DNA in a linear message of nucleotides, like 6 billion nucleotides in every cell of our bodies, to messenger RNAs, a little more complicated than that, but to messenger RNAs, and then to translate that information into proteins. Proteins are like hemoglobin, collagen, enzymes, structural components of cells, and many other functions. And one of the things we're trying to do is to fill in the picture of all the predicted proteins from the genome, how many have actually been identified, detected in biological specimens, in biological functions. And that's what I'm going to tell you about today, and how we used big data approaches to understand how to predict the presence or the likely expression of proteins that have never been detected before, and how to predict and demonstrate the functions of proteins that are readily detected, but for which we do not understand yet their functions in the cells. Okay. This is 2018. 17 years ago, in February, these two covers of Science and Nature magazine. How many of you know about Science magazine? Nature magazine. Okay, so weekend, mid-February, they published the same weekend these two covers. In fact, it was a full issue of sequences of the human genome and articles about the sequencing of the human genome from a public sector group led by the head of the Genome Project in the United States and many others from around the world, including, very importantly, China. The second project was led by a different individual who was in charge of a company-sponsored initiative. And there was a big competition. In the end, they agreed to have it published side by side, separate journals, and to announce their findings together. So there's a good story about cooperation as well as competition in science. Now the reason I show you this is 5 days later was this cartoon in the Financial Times of London, England. Do you see what's going on here? What's in the middle of the cartoon? Anybody? Think of the nature of my talk. Protein, right. So this is a globular, happy, active, dynamic protein. And where is the DNA?
Outside and out on stage.
Over here, off stage. It was just the center of every news item in the world 5 days earlier. Check it off, genome's done, except it's still not done. It's still being annotated and corrected for many challenges. But here's the DNA, off stage, watching the protein. Biotech's next holy grail, that means the next huge challenge. To decipher the human protein set. And that's what our Human Proteome Project is actually addressing. Here's another way to think about it. If you start from the phenotype, that means your face, your smile, your energy, your health. And you work your way back all the way to the genes. So here's the genome, all the genes, and here's the phenotype. Health, disease, affected by the environment, exposures, things we can measure in patients, individual volunteers. Then you go from the population to individual, to organs, tissues, cells. Cells are the center of our workshop this year here. Microbiome, these are the microorganisms, bacteria that live inside our bodies. Here's the metabolome, metabolic pathways, and here's the proteome, sort of in the middle of all this. And here's one kind of RNA, here's another kind of modified RNA and DNA, here's RNA messengers, there's variation in the DNA, and here's the genome. So you can start from the whole animal, the whole person, or you can start from the genome and ask, how do we go from the genes all the way to our phenotype, features? We published this 8 years ago. We just launched a big national center for biomedical computing funded by the National Institutes of Health of the U.S., which was called Center for Integrative Biomedical Informatics. And the key word here is integrative. Integrative means you bring together all these different kinds of studies and factors that affect biological processes. So, how many of you have done any research or reading about proteins? 3. Are you all computer people? Mathematicians? Genomists? Genome people? It's great. Everybody can learn a lot. Here's a big message that from genome to phenotype has to go through the proteome. And Professor Schmidt just reminded me that he has chosen to use some of his precious time and his big brain on this subject of the proteome. It's a worthy challenge. It's welcome, very welcome. Proteins are the major action molecules of cells. Proteins and their many derivatives. So proteins undergo changes. We'll talk about that. Proteins are dynamic. DNA, for the most part, is the same in every cell in the body. You get one copy of each gene from your mother and one copy from the father, and you've got them. They have to be reproduced, of course, when you have more cells made, but there are 2 copies of every gene, essentially every cell. Proteins can have a few copies or a great many copies. It may change dramatically in the course of 24 hours of the day, or from early development and fetal development, newborns through growth and development into late stages of life. Many proteins undergo lots of changes, normal, healthy changes, as well as related to disease. Proteins also play critical roles in regulating the expression, and therefore the function, of genes. So there's all this feedback, very complex. One of the big parts of biology and molecular biology in the last decades has been to try to understand the proteins that are involved in regulating genes and gene expression. Proteins carry out so many important functions in the cells that they're very important targets for developing drugs to treat disease. So they're the primary targets of most pharmaceutical agents. They can be drugs themselves. That's to say, we know how to turn proteins, especially antibody proteins, into drugs that go and attack a particular molecule, like a molecule on the outside of a breast cancer cell. And it's a successful treatment for one important kind of breast cancers in women. We also study proteins because they may be biomarkers, indicators of disease. Maybe also the mediator of the disease process. So if you can figure that out, then you can figure out what needs to be modified to prevent the disease or reverse the disease with treatment. And finally, there's been a tremendous amount of progress in the technology applied to studies of proteins. I don't know if you've had courses on the history of science and technology. Anybody had a course on science and technology, policy, science and technology, history, anything like that? Well, usually the way it's taught is to say you start with basic science, basic research, and then you figure out something practical, applied research. Dr. Schwann here is a master of starting companies that do this. And then you have technology and maybe business. And then the technology can be used to help the research. But it's usually presented as a linear process, basic to applied to technology applications. But what it really is a huge feedback, because new technologies make basic research feasible. In the human genome, sequencing the human genome, 6 billion base pairs, 3 billion in each of the strands of the DNA, took entirely new instruments. And there was one lab especially that developed ultra-fast microsequencers for DNA and microsequencers for proteins, and synthesizers for DNA and synthesizers for proteins. Without those machines, the Human Genome Sequencing Project would have been impossible. In fact, there were most august scientists of the time, 32 years ago, in 1986, when this was decided to proceed, who ridiculed the project, said it's impossible. And if it's possible, it's not interesting. And if it's not interesting, why take the money away from my research to do something so mechanical and repetitive as trying to sequence DNA? Had the same problem for proteins, sequencing proteins, figuring out how to detect proteins in biological specimens like the plasma of the blood or in the fluid from a tumor requires very good techniques, and we now have instruments, reagents, and powerful data analysis methods we call bioinformatics to facilitate that word integration, or a term I mentioned before, integration of multiple kinds of data, multiple techniques to ask big questions. So I've already given you a clue about this. It's worth thinking about all of biology as an information science. You know, it's easy to think about computer sciences and related, machine learning, deep learning. All of that is relevant here, but the biology itself is an information science because there is a coding of information in the linear sequence of DNA. It's organized in 23 pairs of chromosomes inside the cells. How many of you have heard of chromosomes? How many chromosomes do you think you have in each cell? You know? Right. Which everybody should have figured out, because it's 23 pairs. Right? Information goes from a DNA sequence through what we call transcription to make RNA. The RNA undergoes some very complicated, I don't think we should get into this today. I gave a whole lecture about this when I was here 2 years ago. It's fascinating. One of the most remarkable developments of evolution is splicing to make intact gene protein coding sequences from a structure of the gene, which is coding, not coding, coding, not coding. Fascinating, but not today's talk. And then those RNAs, the messenger RNAs that is, have to be translated into proteins. I said in the opening remarks that proteins then undergo other kinds of modifications. They may be cut in specific places to generate two products, or even more than two products. Both may be active doing different things, highly regulated. They may be modified. It is extremely common to put phosphogroups, acetyl groups, sugar groups, up to 200 or 300 or 400 different kinds of chemical modifications on the backbone of the protein. And all of these change the properties of the protein. They're essential to the function. And this is an area that's been studied for a long time, but only now are we getting ways of using complex data from these kinds of studies to understand crosstalk between different changes in the backbone of in the side chain to the backbone of proteins. And the result of all that is a vast number of proteoforms. There's just an article published, big group around the world. How many proteoforms are there? Well, I'm going to show you shortly that there are thought to be now 20,000 protein-coding genes. But we're quite confident that there are, on the average, 50 or 100 different modified forms of the protein for each one of those protein-coding genes, protein. That makes 1 million or more. And that's the complexity of proteins. So why can't we just study the gene, the genome, instead of having to get into this complexity of the proteins? Many people spend their lives just studying the DNA and changes in the DNA, mutations from exposures to the environment, regulatory changes in the DNA. Turns out there are a lot of things that we want to know about gene functioning that we cannot study in the DNA. We must study at the level of the proteins. Some things are in the DNA that say mutations or variants in the sequence, common variants. It's a subject all unto itself. But there's no information that we can get from the DNA that tells us about the dynamics of protein concentrations, as I said, during the day, during life development and disease, during exercise, during menstruation, and many, many other normal functions. The DNA gives us no clue about the post-translational chemical modifications that define many protein functions. And it also does not tell us about the splice isoforms. So this is a big subject. Proteogenomics is a word new word, recent years, that brings together proteins and the genes, which is sort of the subject of this workshop and this particular talk in this workshop. The importance of this one slide is that we can learn a lot of the fine detail about genes that we really don't know yet from studying the protein products. So protein information feeds back on understanding the genome. And understanding the transcripts gives us a lot of information about where to look for proteins being expressed and how the proteins might be modified and other useful information. There's even, this gets very complicated very fast, I'm sorry, but just so you should hear it, for many years there was thought that most of the DNA didn't have any functions that we knew about, so it was called junk DNA. As if just because we humans don't know how it works, it doesn't have any value. In evolution, we would not expect the DNA to have survived through all the species and all the eons to today without value and function for survival of species. So, we now know that about 1% of the DNA codes for all these 20,000 proteins. When we started the genome sequencing project, it was confidently predicted that there would be 50,000 or 100,000 or more protein coding genes in the DNA. When those two covers appeared, it was down to 35,000. And soon after, it kept dropping. We're now down to 20,000 protein coding genes. But in the meantime, we learned that each protein coding gene has about 5 of these splice isoforms. So that's times 5. So 20,000 times 5 is sort of like the original expectation of 100,000, except that it's in these two different processes, the evolution of the genes and then the evolution of the splice variants. What about the other 99% of the DNA? So it turns out there are RNAs which were quickly labeled non-protein coding, non-coding long RNAs, or long non-coding RNAs, link RNAs. And then there's short RNAs called SMRFs, small or short open reading frames. Open reading frames means in the dynamics of the DNA, sort of the breathing of the DNA and the chromosome and the histones, you can actually see units that may be coding for RNAs. And if they make RNAs, maybe the RNAs could be translated to make proteins. And I think that may be beyond us for today. Just that you've heard it, there are questions about whether there are additional peptides, polypeptides, proteins from these different kinds of RNAs that are detected now coming from the rest of the genome. The rest, after the 1%, it codes more confidently for proteins. If you hear a talk about this, you should be suspicious about what they tell you, because it's very hard experimentally at this point. There are a lot of false positive identifications, which means it looks like you found something that was never found before, but it's really just a common variant of a common protein. It's peptides. Or you have to do such large search space to look for many, many other features that you lose control of the quality of the analysis. And even more basic than that, since we relied on something called mass spectrometry to see the spectra generated when you excite peptides, a lot of people who publish in this area are not experts about reading the mass spectra, and when an expert looks at them and says these spectra don't show what they claim, you realize you can't make a good case from terrible evidence. You have to have standards and guidelines and follow them in a field, and that's what we've done a lot of in the last 15 years to enhance proteomics, quite a lot. All right, now, a brief introduction of the Human Proteome Project. Now I'll tell you about some new things that are a little complicated, but hopefully I can give you an overall picture so you can think about this in context of other things you'll learn about in this workshop. We have two big goals for the Human Proteome Project. One is to make sure when people study genes and cells, or genomes, cells, and mathematics, that they don't forget about the proteins. That's what I've been arguing for the whole talk already, okay? So we want proteomics to be part of the multi-omics, multiple omics integrated studies of genes, RNAs, proteins, functions. Part of that means making the assays more attractive, feasible for people who are not experts in the field. This is a challenge in every field of science, to help people use methods they don't know a lot about with enough confidence and enough competence to ask the questions they want to study. That's what we've been working on for life sciences and biomedicine. The other is up to us, the whole world working on proteins, to complete what we call a protein parts list. So if there are 20,000 protein coding genes, we'd like to know that a protein has actually been detected from that presumed protein coding gene. I'll give you the numbers on that in a second. We'd like to find at least one product, one protein from each of those genes. We'd like to be able to say in which tissues of the body, in humans as well as other species, is that protein detectable and functioning. And then what does it actually do? Characterize it, annotate its functions in a way that gives meaning to the function of the whole cell. And when we know that there are all these other modifications, what are called post-translational modifications, PTM, single nucleotide polymorphisms, these are common variants in the sequence from the mutations, and the splice isoforms. We'd like to characterize as many of those as possible. That is a challenge without an end. There's always going to be more to discover, because there are changes and new kinds of exposures, new kinds of mutations that will continue to build the variety of proteins forever. But we'll build up a much, much greater understanding if we have an organized framework to find what we're doing. So we're going to find the proteins that haven't been found to be detected yet, and we want to characterize known proteins, well-known proteins, for which function is yet to be provided. And we also like to know how many proteins of the predicted proteins are really not going to be detectable at all, given the methods we currently have. We've made a lot of progress on all of these. So here is this Human Proteome Project. Started discussion much earlier, but really launched 8 years ago. And you can think about it this way. There are three pillars. Mass spectrometry, this is measurement of peptides from proteins. Antibodies, this is a way of detecting the surface features of proteins. Whoops. And knowledge base, bioinformatics, databases. This is no small feat. We've gotten essentially the whole community of scientists working on proteins all over the world to contribute their data, their primary data, they're actually measured in their instruments, to a resource, and then we have a registration of the data sets that have been produced and put into repositories so that you can actually go and see what's there. And two of our big partners in this activity actually take all that raw data, run it through their own separate pipelines, and by uniform methods verify what the investigators reported or modify what the investigators thought they found to meet a high common standard. And this has been a huge progress for the field. And originally, we had biology-driven projects, so cancers, cardiovascular, brain, liver, lung, kidney, etc. I was in charge from 2002 for a project on human blood plasma proteins. And we had adopt a chromosome. We said there are 23 pairs of chromosomes. So each chromosome, typically a different country group of scientists or a region like South Asia, joined together and took on responsibility for one chromosome. And we have these different groups. So we have about 50 groups altogether. We have 23 chromosome groups, actually 24 because there's X and Y for the sex chromosomes, you probably know that. And there's mitochondria. And then there are 22 groups for biological processes and disease categories. And then there are these three pillars to which we've now added pathology. The data start with the investigators, individual investigators all over the world, and they put their data into one of these resources under the Registration and Proteome Exchange, which we created starting in 2008. We have metrics that apply to evaluating progress, publications, many dozens of publications, 164 in one journal of proteome research over the last 5 years. I mentioned already we produce guidelines that govern the publication of data and the acceptance of primary results. And all this is brought together under a resource for peptides with mass spectrometry and curation of all information, not just by mass spectrometry, but by antibodies, by protein sequencing, by three-dimensional structures of proteins, other kinds of biochemical and genetic methods. So we basically covered this. We announced the global project. We've been putting it out in publications and in open source shared data. We've had a major effort around data sharing and quality assurance. We've developed a way of doing targeted proteomics. This is a whole special subject. Instead of just measuring all that you can in a given sample, where you keep measuring the ones you've already found before over and over, you can target the mass spectrometry to just recognize the ones you want to measure and quantitate. And a very special feature of proteomics is that it's not just detecting yes or no the presence of the protein, it's quantitating. And for many studies in biology, it's very important to know how much and how much something changes. And for mathematical modeling of biological processes, quantitation is extremely important. Come back to this. So I have three themes this talk. First, to build on what I just introduced by telling you the progress we've made on the HPP, Human Proteome Project metrics. Then I want to give you, the time available, a couple of deep dives on using computational methods and big data approaches to answer two challenges that we've set ourselves about finding proteins that have never been found before to be expressed and predicting functions of proteins that are really easy to measure in some cases but haven't been characterized for their function. All right, this covers 6 years of work right here in one slide. What you need to know is, and this comes from Nextprot. I was in a slide with the workflow. Nextprot is a service provided by a big consortium in Geneva, Switzerland. It is our gold standard database for curated protein findings. And what they do is they classify proteins according to the approximately 20,000 predicted protein coding genes for how many have actually been reliably, credibly detected with protein methods. So you can see here, by 2012, we already had almost 14,000. It's a pretty good start. That's like 70% of the goal. And it's been moving up. This is the latest number earlier this year, 17,470. That is 89% of these first four categories. So 89%, that's like, you know, a few steps to the finish line. Except it gets harder and harder the more you've found to find the ones that have been resistant to being found. Okay? The second line is very important. These have not been found, or not found with sufficient evidence, as proteins in any tissue, in any organism. we do know that the RNA transcript, the messenger RNA for that predicted protein has been found. Following? Remember, DNA, messenger RNA, makes protein. So if you know that the messenger RNA is expressed in the liver, or in the testis, or in someplace, in the back of the brain, if you know where its transcript is most expressed, it's a pretty good clue where you should look to see if you could find the protein. Now, why wouldn't you find the protein? Well, there are a lot of reasons. One reason is it may not have any sequence features that can be detected with our mass spectrometry methods. Just to understand, the way we do this is we use a particular enzyme that cuts proteins in a certain way, releases peptides from the length of the protein that then fly in the mass spectrometer upon laser excitation, getting space age stuff here, and can be recognized. There are a lot of reasons why that doesn't happen. If you don't have any sequence site that this enzyme cleaves, you can't use it by this method. Can't detect it by this method. If it's a protein that's embedded in membranes and isn't solubilized, doesn't come into the solution from the tissue, and you can't reach it with the enzymes, you can't detect those. It may be that the protein is a close family relative of other proteins that do similar things, or maybe different things that have very similar sequences, and therefore we can't tell the sequences apart. If we can't tell them apart, we can't count them individually. Yeah? If the protein isn't made, it's not detectable. If the protein is made, but at a very low level, maybe because it's degraded, proteins undergo synthesis and degradation. So some are degraded such that their actual level is too low for the methods that we have available. There's always a question in any kind of detection method for anything, chemicals, proteins, genes. What is the limit of detection? How low can you go and still detect it? So for all those reasons, we may not be able to detect it at the protein level. But if it's not detectable at the transcript level, if we can't find any tissue where the transcript is expressed, we don't have much chance of finding the protein. Maybe it's not impossible. Maybe the transcript itself is degraded rapidly. And there's a whole process about that. But anyway, that's the transcript. The next category is much less common. It's from other species. Good data in other species, but not in humans. Well, in other species, if it's made in part of the brain or it's made in the heart, we could take that information and look in humans. And we do that. And finally, there's a category of modeling. This category is an embarrassment. This is from the gene level. When it says uncertain or dubious, it's about the gene being the protein coding gene. It's not about the protein evidence. And for a variety of reasons, these are a small class, but 574 is a lot when you're trying to get down to the last 2,000. And we decided 5 years ago to exclude category 5 because we don't think the problem there is protein. We think the problem there is it's not really a protein coding gene. This is what I meant also in the beginning when I said studying the proteins leads us to understand a lot more about what we don't know still about the genes and the genome, and this is one big category. So, you might ask, of the proteins, the 17,000 now, and here is the summation of these is 2,186. Those are still missing. That's a big number, especially if you're looking for one at a time. It could take a long time. Well, this is what we accomplished, the world has accomplished, not just our group, in the last year. There's momentum here. From January 2017 to January 2018, the PE1, this is protein evidence, went from 17,008 to 17,470. I don't know if you think that's a huge gain or not. However, it is 462 newly identified PE1 highest credibility protein identifications. Where did they come from? Well, 431 of them came from the PE2, 3, 4, almost all, from knowing where the transcripts were present, you see? And I'll show you something about that in a second. And there were some other details about the rest, but none of them came from this PE5 category, which I'll try to put out of sight. Here's another way to look at what we have. The 17,440 is the sum of the green and the yellow. So the green is by mass spectrometry. This is the workhorse technique of proteomics, mass spectrometry. But it's not trivial. 1,378 are by non-mass spectrometry methods. And here's what they are. 530 by protein-protein interaction studies. That's a whole growing class of studies, quite separate from mass spectrometry, mostly separate. There's about, there's 99 by sequencing the proteins. There's 170 by studying the post-translational modifications and going from that to figure out what the proteins, just say, some by mutations, some by 3D, three-dimensional X-ray structures of the proteins, some by antibodies, and many other biochemical studies. Please.
The disease mutations, would they be cancers?
Well, they could be, but there are many, many other kinds of diseases where there are specific single mutations that cause growth and development abnormalities, heart disease, every kind of disease. It's a great variety. Cancers are a little different from others. They have many mutations, but they're typically not in the germline. So for this purpose, it's equally good, actually, because if you can show mutated protein from a cancer, you can still study the protein from the cancer. So it works both ways for this particular application. Good question. Any other questions? All right. And then the rest of the categories are the ones that are missing, plus the dark wedge for the dubious. Now, you can say, well, are we still making progress with the mass spectrometry? Because about 2-3 years ago, we published that maybe we're saturating, we're up to the limit with mass spectrometry. We've done a lot of different tissues, some field samples even, diseases. But you can see here we're still finding more mass spectrometry. And the big reason this past year was post-translational modifications. This is a very unusual class, not studied much, but it's opened up a whole another set of proteins that wasn't picked up by standard methods. Here's another way to think about it. What about these families of proteins? I said before, there are families that from evolution, where you have a duplication of a gene, duplication again, then each gene evolves further, develops new functions or modified functions, and there are large families. So look at this. There are what's called olfactory receptors, sense of smell. Okay? There are at least 400 genes in all of our cells for receptors for different smell chemistries. And almost none of them have been detected. This is a very hard problem. First of all, they're not as important, we think, in humans as in rodents and other species. Yes?
Is there a distinguished difference between the human olfactory receptors and the dogs or other animals? Have we done any experiments to detect whether those proteins in other animals there are?
Yeah. So the question is, what do we know about olfactory receptors in other species? There's a lot known, because in other species, animals that sniff a lot and whose behaviors, reproductive behaviors, aggressive behaviors are highly influenced by what they smell and the scents that they lay down, the smelling chemistries that they pee on the soil or whatever. It's a big part of their lives, and it's part of our evolutionary history, and a lot of people are studying this intensively. Even in other species, these are hard molecules to detect. And the reason is that most olfactory receptors are in a certain region in the brain, I've come to think that they are produced in neurons by cloning. Let's say one clone of cells, just a few cells make this particular smell specialty, and another clone of cells makes another special kind of smell detection receptor, and there are hundreds of these, but each one is hard to detect because either they're not producing much RNA transcripts, which is true, or transcripts are not being translated into proteins, or if they are, the levels are too low, and the cellular samples are too hard to get. For a long time, we didn't have really any samples of human olfactory epithelium. It's in the base of the brain, in the front of the brain. Very hard place to get. It's a pure volunteer, those samples. But even when they've been studied finally, it's very hard to detect expression at the protein level, and it's even hard to detect expression at the transcripts level. They must be at low levels or be turning over very fast, or both. Anyhow, as you can see, there is no progress on finding those in the last few years. But look at these other ones. These are other membranes. I told you membrane containing proteins embedded in membranes are hard to get into solution and measure, but we have made progress. There are new ways of doing it. It's making progress. Here's another kind of protein. It's called zinc finger protein. It's very important in regulating genes. Look at that. The numbers decreasing are the ones that are missing. So the more we find, the lower this goes. Same thing here, same thing here, same thing here. All these different proteins, all 5 of these big classes, there's been a lot of progress. But the olfactory receptor specifically still resistant. So it's an opportunity. And there are some groups around the world particularly working on this. Still have ways to go. So last year, in our special issue from participating investigators in the HPP, Human Proteome Project, we had these novel approaches. This group in Shenzhen, just up the road here, used a solubilizer to get membrane proteins, and they used these beads. This is very clever. These are hexapeptide beads that absorb proteins. And once they absorb their capacity, you can't force more protein onto the bead. So if some proteins are present in large amounts and others in small amounts, small concentrations, the large amounts, they'll fill up their beads, but then the rest just washes away because they didn't bind. It's a saturated bead. And the less abundant proteins will fill up, or maybe not even quite fill up their beads, but relative to the more abundant proteins that are usually blocking us from detecting these, they're enriched. Follow this argument? This is a general concept of how do you enrich for something that's hard to find. One way is to get rid of the stuff that's too abundant, that interferes with the detection. And that's what abundant proteins do. So they reported quite a few candidates, but 15 of them they were able to verify by a separate method I don't need to go into. And then this group carried forward third year of their studies on sperm. They've found almost 300 missing proteins now in sperm. This is amazing. And it all came from knowing the transcript information that sperm and testis are rich in proteins and transcripts expressed nowhere else. I'll show you a slide of that in a minute. And in Guangzhou, I think, they used multiple enzymes, not just the one enzyme we always use, which is trypsin. And this group in China studied the kidney and looked at not just the proteins, but the phosphoproteome. This is where you have phosphate post-translational PTMs on the group. And it's very important for protein signaling in the metabolism of the cell. These folks in Iran study fetal development of the heart. And they're interested in Y chromosome. And they had one protein that they very cleverly showed that they had detected, hadn't been detected, isolated before. So all those, so out of these 15, when the whole data set was examined, and some that they hadn't validated themselves, was put together information, we got 20 new findings here. 4 out of these 12, this is incomplete, we're still looking for more spectra to validate those. One of these, this one. Now this is very interesting. This is maybe pushing it pretty hard on my mass spectrometry, but our rule is that you can't claim a protein as identified with just one peptide. The risk that could be a peptide from another protein is very high. But if you have 2 peptides of at least 9 amino acids each, in the old days people used to use 7 or 6. And the shorter the peptides, the more promiscuous they are, matching to many different proteins. So these guys, what they did in Japan was do an informatics exercise. They looked in the databases, they're available to everybody, and said how many of these have at least one peptide for a missing protein. And they found about 400 like this. And out of those, they looked to see if they could find in the same database or no database, but from different investigators, different data sets, contributed to our now open source proteomics information, to see if we could find a second peptide of high quality that would make the case that 2 different peptides from 2 different labs in 2 different repositories actually could be brought together informatically to show that they match up and produce finding. So I call this the stranded peptides strategy. These peptides are out there in the databases, but nobody actually gone to the trouble to try to pull them together and try to verify them. So they proposed 41, and that was cautiously, I would say, we published that in our journal. And of those, 16 were curated by Nextpro, but I should admit that they weren't based on their data. They didn't know what to do with these data. We were having a policy discussion about that because the question is how reliable is the information? Where do you actually get the confirmation? What's the provenance of the samples? Many good questions. But pulling all the data and other data together, 16 were validated. So the result was 43 new PE1 proteins, 18 from other sources of data, as 43 as part of the 462 that were added last year in Nextpro. Okay. Now, as time runs on, I want to get one more topic before you. And the transition is this. The collaborating on searches for these missing proteins, and next on the uncharacterized, well-established proteins lacking functional annotation, is a good strategy. More and more in science, we want to collaborate. We want to bring people together who have different expertise, different knowledge, different driving questions, and show that you can actually, the combined effort is much more productive than just everybody on their own trying to win small victories. You can do big projects if you can get much more cooperation and collaboration. So here's a little bit about chromosomes. One of the 24 chromosome groups I told you about is the one that I've been reading for a number of years, chromosome 17. We picked it because we're interested in breast cancers, and TP53 is a very important cancer gene on chromosome 17. Breast cancer 1 gene is on chromosome 17. ERB2 is on chromosome 17. There are a lot of breast cancer genes on chromosome 17. And we've done a lot of interesting information. This chromosome also has a whole bunch of olfactory receptor genes, and they're in one area right there. It has keratin and keratin-associated proteins. 53 of these in this one area of the chromosome here, and so forth. It has this ERB2 amplicon, 21 genes around a gene that's very, very important in breast cancer, and some of them are expressed together in HER2/neu ERB2 breast cancer. Oops. Okay, so the question is, how much progress have we made on missing proteins in chromosome 17? The answer is, since the challenge to find 50 new missing proteins in 2016, our chromosome has gone from 148 missing to 105. That's a gain of 43. That's pretty good. And we didn't do it all. I mean, hardly did any of it, actually. But we got all the information together from everywhere and brought it to light. And we've now analyzed very carefully which of the 43 were PE2, this is transcript information. Okay, see, these were missing proteins. None of them were PE1. So all the ones in this, let's call it red color, were PE2. Those are good prospects. And these have at least one peptide available in mass spectrometry. And these were found by protein-protein interactions. So we did a detailed analysis of which proteins, what kinds of proteins were found by mass spectrometry and why, and which kinds were found by protein-protein interactions and why. And this lays out the plan for experimentally finding the next ones, because these have now been upgraded. But we still have 105 left to find. And something similar to this was done for chromosome 2 and chromosome 14 by the Swiss-French consortium, which focused on the testis. At the same time, the antibodies group in Stockholm, another part of our program, showed that more than any other tissue type in the body, male tissue, so-called, testis, and associated organs, was rich in proteins found nowhere else or only a much lower abundance in other organs. And after that, brain. So these I've mentioned a couple times, testis and brain, the rich sources of finding previously missing proteins. And we went through and looked at all the properties for mass spectrometry, properties for protein-protein interactions, and we've laid out a strategy for how to find some of these 105 still missing. We're running out of time. I don't want to go through this in detail, but I'm leaving my whole slides there. If any of you are interested enough about this to try to understand this in detail, you can work through these slides, and these papers will be published in the next several weeks. They're not published yet. This is unpublished data I'm showing you here. I think it would be a very interesting little project as an individual, as a group, or as a student class activity. And we've also used the stranded peptide approach. We found one that is beautiful. This is what we found in the databases compared to a synthetic peptide, which lines up absolutely perfectly on the mass spectrometry. And 6 others that are prospects but don't match up nearly as well. And we also did this kind of analysis for the proteins that were found by protein-protein interactions. How were they found? What were their properties? And we looked at the ones remaining to pick out star candidates that could be found with mass spectrum, with protein-protein interaction, and to suggest what interaction proteins would be most effective for those missing proteins. And here's the summation. So 105 we still need to find. There are 10. We already have one proteotypic peptide. We just need to get the second one. And we have 15 that don't have a peptide to start with yet, but we think are feasible. And down here, including 4 olfactory receptors that have some prospects because there are new ways of looking for them with protein-protein interactions. Okay, finally, I said it was very important to figure out the functions. This is the connection between genes, genome, and cells. Is anybody coming in on a dot for the next class? Is next class coming in? Is there a next class? Are you want to go another 5 minutes or so on this? Okay. So this is a very remarkable story here. There's a colleague of Indika's and mine at the University of Michigan named Yang Zhang, came originally from Wuhan. He was a theoretical physicist, and he won a prestigious Helmholtz Fellowship after his PhD to go to Germany, came back to Peking University, pointing to the faculty in physics, decided no, he'd rather study proteins. And he started figuring out how to use computational methods to predict the folding of proteins, the functions of proteins, the docking of ligands to proteins, and many algorithms for protein folding, structure, function. So I went to him with this question, which is, could he help us figure out the likely functions of these PE1, these are well-captured, well-identified proteins, but uncharacterized for function, and predict the function. Okay? So we put a really good graduate student on this, Chengzhen Zhang, and this work is now done and submitted, and I'm proud to show it to you today. So the basic idea here is to use algorithms that take any protein sequence and look for homologies. These are sequence homologs, similar sequences and what their information is. And to look at the templates through threading algorithms that are complicated for the structures, three-dimensional structures. And then to combine with protein-protein interactions using a string database. And from all of those we get predictions of what's called gene ontology terms that I'll show you in the next slide. Now, this is chromosome 17. And if you want to study this, I have a set of slides about the ITaser at the end of the, past the end of the talk. I also have this one, which is a little more detailed, but I just showed you. And this is the way ITaser works. It has a threading algorithm for the folding based upon the amino acid sequence and energy minima. So there's a structure assembly, a clustering mechanism, and there's a reassembly and a full atomic refinement with several tools here. And then we'll use the coach and cofactor algorithms to match up function with structure. And here's a little more detail of the same. We want to understand ligand binding. We want to understand any enzyme activity predictions. We want to know for gene ontology, what is the biological process? What is the molecular function? What is the cellular localization inside the cell? Because proteins do very different things in different places in the cell. The inside of the cell is a very complicated, interesting environment. There's the nucleus, and the mitochondria, and the Golgi, and membranes, and so forth. So what's shown in this slide is we started with chromosome 17 has 1,166 protein coding genes. And there are 105 now that are still missing, but there are 66 that are unannotated that are PE1. Out of 1,166, there's 1,038 that are PE1, well-identified, credibly identified proteins, but 66 we don't know the functions. So they looked here with several common methods, and I got modest prediction of functional terms. But when they used their combination of ITaser and cofactor, using, let's take the green here, or here's the green, this is protein-protein interaction, red is sequence, blue is structure, and black is the combination. And you can see for all three of these kinds of function-related terms, the highest performance by a good margin is the combined cofactor approach. And of the cofactor, the factor that contributes the most is the structure. These are novel findings. And here we tried to find out where's the maximum. It's not a very sharp maximum, as you can see, but here we drop this down, and here's the confidence score, the C-score, for these three different approaches: molecular function, biological process, cellular component. Here's an example. Famous protein TP53. A lot is known about this. This is a positive control. And we know there's DNA damage response, regulation of cell cycle, very interesting to indica. And if you compare the protein in human, with the protein in the worm, C. elegans, very much studied model organism. If you look carefully at this, they are very, very similar. And they can show it actually binding to the appropriate part of the DNA to carry out these functions. And they predict the same gene ontology terms for both species. Okay, so up to now what we've done is shown that our tools work on known, well-annotated proteins. Okay? This was on the, maybe I didn't say it clearly, 100 randomly selected PE1 chromosomes that are already well annotated for function. So we know that the method works pretty well. Now we're going to try it on ones that don't have any functional annotation and see how many we can generate good predictions. And the answer is, a lot depends on where we draw the cut points. But with the values that were derived from the previous study, the positive controls, we would think we've got 13 by molecular function, 33 by biological process, and 49 by cellular localization out of the 66. This is quite amazing because the rest of the world is trying, proteomics world is trying to do this, and they are studying 1 or 2 proteins at a time. It can take 2 years, 3 years. Isolate the protein, study this, study that, make changes, knock down the protein, see what happens to the cells. And there was just a paper published, I was even a co-author from Korea, a very nice study, 1 protein, several years of work, including model findings on C. elegans, and it's a credible report. But here, with this rapid and powerful algorithm, we believe we've given direction for ourselves and for others how you could rationally look for these kinds of findings. And the examples are here. So here are the 13 highest rated by molecular function, and I've started a couple. This first one and the fourth one both have extremely high scores for transferase activity. Well, you could say that's not very specific. But in fact, it's for sphingosine and acyl transferase. This is a very important function in the lipids in the brain. And it's true for this one down here also. So this would be a terrific target for experimental investigation. And then here's another one. This is a cytoskeletal binding protein, we say, and we think it binds spectrin, which is important. And then if you look at this in three-dimensional structure, it actually predicts a structure which matches very closely an E. coli protein for sugar transmembrane transporter. So this gives a huge boost to looking rationally for functions of these proteins. So the summation of all this is the metrics. We made a lot of progress. We're now at 89% of total predicted proteins and 15,798 canonical in the peptide atlas. There's still 2,186 missing proteins. For our chromosome 17, we are very close to the next 50 missing proteins to be found. We've already got 43, plus the excellent mass spectrometry stranded peptide I showed you, plus 35 high priority PPI or MS targets, specifically identified. And finally, we have at least 13, maybe up to 33, molecular function and biological process, uncharacterized PE1 chromosome 17 proteins with now high confidence functional annotations using our predictive methods. So I hope I've given you a sense of the broad picture of DNA, RNA, and proteins, the essential role of proteins in going from genomes to cells, and enough examples of computation and the underlying mathematics, which is embedded in all of that, to make a contribution to the theme of this workshop. Thank you very much.
Any questions? And also,
Is there a way to take all this research and, say, increase the human lifespan?
Well, that's a good big question. I'm a member of the National Academy of Medicine, National Academy of Sciences. We have just launched a initiative with all the new excitement in molecular biology and many things, not molecular only, how to be healthier from an early age, from before you're born, to have healthy features, exercise and good diet and avoid nasty exposures and try to avoid the diseases that cut lives short quite dramatically. But the goal is not just to live forever, because the quality of life is so important. So our goal is to enhance the healthy lifespan of humans. And there's going to be a competition, big prizes. People come up with ideas and to get some moderate funding for promising experimental approaches. And eventually, there are a big number of prizes we anticipate for people who actually show they can make a big difference in some feature or other related to healthy aging. There's a lot of interest, and donors are interested in this. It's not a big surprise. And it's a little like the X Prize, for private sector space flight. So we'll see how that plays out. In the meantime, we have pressing problems dealing with diseases that are taking a horrible toll late in life, like Alzheimer's disease, neurodegenerative disease that impair cognitive functioning, thinking, making decisions, being aware of your emotions and your intellectual challenges. In the United States and all around the world now, this is an epidemic of degenerative brain disease, and figuring out ways to, fresh ways, to understand the disease process, to recognize almost surely that there are many different causes of what look like similar patient problems, and to find ways to intervene either earlier in the process or moderate the progression once it's started. All of this is a big subject in the United States and all around the world. And in principle, this kind of basic research will play into all of that, of course. There's a new technique called gene editing, CRISPR-Cas9, previously zinc fingers and talons. There's a succession of these molecular methods enabling to change certain genes, maybe to correct a mistake in a gene, a mutation in a gene. This is all promising, but early stage. Yes?
Yes. Hello, Professor. Because I haven't done any research in this kind of area, so maybe my questions are done this very we know that we have like a gene regulation network, which maybe they're in progress, but we have that kind of network. So I'm wondering whether we have the protein interaction networks, and if we have, like how good are these? Can we utilize them to help us to identify cell groups or to do like clustering or something?
Yeah, that's a wonderful question. Questions about networks. So biological functions, I appreciate you bringing this up, are organized in pathways and networks. So pathways, for example, if you look at metabolism, if you eat a sugar molecule like glucose, it goes through a metabolic pathway step by step. We know every detail. It was brilliant research of 2 generations ago. Still a lot to learn. If you ask about the pathways in the brain affecting thinking and Alzheimer's, we don't understand nearly enough about those pathways. Networks are a very commonly used way to explain interactions of proteins, so these protein-protein interactions I've been talking about, protein-lipid interactions, protein-nucleic acid interactions, and you can ask how well connected are the activities of 2 different proteins, or the isoforms, like the splice isoforms, of the same protein or of different proteins. So in fact, one of our postdocs at Michigan from South China published a whole series of papers with me on this, on the genome-wide, proteome-wide prediction of networks at the isoform level. At the gene level, I think is a little unimprecise, because we know that the gene is a mixture of these different transcripts produced by splicing and their proteins. So we think this is a major advance, but it needs to be validated experimentally. And there's been a lot of interest about this. Yes, indeed. If you know the term systems biology, networks and pathways are the heart of systems biology. Good question.
Okay, I think, Michael.
Thank you. Thank you for your talk.
How is the human protein project finding and characterizing, helping and affecting the drug discovery, new drug discovery? New drug.
For new drugs, yeah. I think the fairest answer is to say we are not well connected with new drug discovery. However, we are providing a substrate for the biotech and pharmaceutical companies to have much more information for new drug discovery. The biomarker work is at the heart of what the drug companies need. They need to have clear evidence of what the target should be for a drug that could be designed or could be used from previous work to modify breast cancer, modify Alzheimer's, modify blood pressure, and this is a vast enterprise. There are several proteomics scientists at big companies like Roche, GSK, and others that have participated in the HPP. So they bring their own interests, and we're always welcome and we're very eager to have company participation. But usually they're reluctant to share their findings. So, we'll work with anybody, but the goal of this is open source, open sharing, maximal progression of the field, and we're thrilled if anybody can turn it into benefit for patients. We don't have good examples yet.
I think we have tea outside. There are a lot of good questions, and Professor Omen will be here till next Tuesday. And then please make appointment with him, and you can discuss more good questions.
Actually, only until Monday morning. Early Monday morning. So you have to catch me by Friday.
So you know his email number, you can write to him and make appointment. And this is, we are highly encouraged that. And that's why this is month long and they come and stay for a week.
Well, I'll stay around the rest of the afternoon. Anybody while it's all fresh, is a good time to think about your questions. I'm happy to have emails or follow up. I'll be around tomorrow afternoon, Thursday afternoon. I don't think we'll have anything here on Friday, do we?
No. So you guys have a lot of time in the morning.
Catch me when you would like, but catch me most of all right now.
Yeah.
Great. Thank you all for coming and staying.
Yeah. Yes, small senior from CTU.
Okay, thank you.
So I'm. Okay. Thank you.
Okay.
Thank you.
All right.